Today we're going to be discussing motor systems. So this is how our brain controls our muscles. This is the first in a series of lectures on motor systems, and today is going to be the major overview of how our brain controls our muscles. And then we will talk about more specialized systems in the next few lectures. And so this entire second portion of the course is on motor control. So at this point, we have learned in great detail how our body perceives the world through sensory information. Sensory information is picked up at specialized receptors, in this case we're illustrating receptors in the skin, and is relayed up into the brain. It's transmitted up into the brain where there's processing and perception that occurs up in the more specialized sensory areas, such as in sensory cortical areas. We have learned about how there are sensory neurons in the cortex that do this. Information gets up to the brain through the dorsal root ganglia into the spinal cord, originating with these specialized receptors. For the rest of the course, and for the rest of the second portion of the course, we're going to be discussing how motor systems control muscles. So that is essentially the outflow from the brain. Information comes into the brain, it's processed, and then the body has to react. That's going to be the motor portion that we're going to discuss. So we will be talking about pyramidal neurons that are in the motor cortex. We will be talking about motor neurons that live in the ventral horn of the spinal cord to control our muscles, the synapse onto the muscles. And we will be discussing neuromuscular junctions and the muscle fibers that. So there are three principal types of movement that motor systems do. First is reflexes. So these are the rapid, stereotyped, involuntary movements that are always the same over and over again, that have very little input from consciousness. You don't really think about these things. One of the classic types of reflexes is the knee-jerk reflex. We'll discuss how that works and the simple but elegant system that does that in a little bit more detail in a bit. There's also rhythmic motor patterns. These are initiated consciously, voluntarily, and then you can of course stop them as well. They are maintained though through reflexes. You don't, when we initiate, say, walking, which is a rhythmic motor pattern, you want to walk, so you start walking, but you don't necessarily think about the details of walking. You don't think about swinging my right leg, swinging my left leg. It's an entirely maintained reflection type process. We're not actually going to talk about this in great detail because it's a relatively complicated system and it's a little bit beyond the scope, but we're going to cover it a little bit. And then, of course, there are voluntary movements. So these are purposeful, goal-directed, and learned movements. They improve through practice. We're going to spend a lot of time talking about this, some of it today, but in the next several lectures, we're actually going to talk about how the brain directs goal-directed behavior and learns goal-directed voluntary movements. So the motor system, it has kind of it can be broken down into three levels of how motor control occurs. The first is the cortex. So this is the place where planning of activities and issuing general commands occurs. Even a fair amount of non-voluntary information is initiated in the cortex as well, although the entirely voluntary information, all of that originates in the cortex. The brainstem is a critical area with several different levels of nuclei that regulate all kinds of neural activity. It synapses onto the spinal cord as well as the muscles of the face, head, and neck. A lot of involuntary reflexes are regulated by the brainstem. And then we have the spinal cord, where there can be coordination of activity occurring just within the spinal cord, completely independent of the cortex. So the knee-jerk reflex, for instance, that originates in the spinal cord. And if obviously you can see that the motor cortex and the brainstem all have to talk to the spinal cord in order to initiate the muscles of the body to move and react. There are other areas that are critical for how motor systems work. The basal ganglia and the cerebellum, they run parallel to the cortex, and there are several loops involved in this process. They are used for the learning and coordination of planned movements and learned movements. We will have separate lectures on the basal ganglia and the cerebellum, so we're not going to discuss those today. And other important points are that each component of the motor hierarchy contains a somatotopic map of the body. So this is similar to how the somatosensory cortex has a somatotopic map. Each of these areas also has somatotopy. So there are additional maps in how they are organized. And we'll talk about some of that organization in a bit. Each level has, it can receive sensory information that can modify descending commands. And the higher centers can modify both sensory input and spinal and brain stem reflexes as well. So just because there are reflexes that occur, there can be modification that happens from the higher motor control areas, like in the primary motor cortex. The motor control systems can also be broken into two additional major levels, upper and lower motor neurons. Upper motor neurons, they reside in higher motor centers, such as in the cortex. This is an example of an upper motor neuron here. It will regulate movement by influencing lower motor neuron circuits in the brainstem and then the spinal cord. So you can see that this neuron can synapse to motor neurons that, say, control muscles in the face, and it can go all the way down into the spinal cord, say even to down into the sacral areas of the spinal cord to control the motor units in the that innervate, say, muscles of the legs. So these are very long neurons. Cell bodies that reside in the brainstem or cortex and axons, they descend and synapse onto these local circuits, and then onto lower motor neurons. So lower motor neurons are in the spinal cord and in the brainstem. So that's referring to these neurons here. These neurons directly innervate skeletal muscles. They can, as we mentioned in the previous slide, be influenced by the activity in the motor cortex to provide for voluntary motor control or modification of ongoing reflex or repetitive movements. That's what these neurons would be doing. But there are also local circuits that can help control and initiate how these neurons fire. And those are the reflexes or repetitive movements. So this is a cross-section of the spinal cord shown here. This is the dorsal side, this is the ventral side. We have already spent a lot of time learning about the dorsal side, where we have sensory information, say from touch or pain, coming through the dorsal nerve. And the dorsal nerve, remember, has this large bump on it called the dorsal root ganglion. That's where the cell bodies of those neurons exist. And it will either say if this is there's just showing a pain neuron, would be synapsing on neurons within the dorsal horn, and that this is generally going to be where sensory information is residing. So we've spent a fair amount of time talking about this. These areas are the various tracts that run up and down the spinal cord. This would be the white matter areas. And then in the center would be gray matter. So these would be the cell bodies of various neurons. Sensory being here. And there is also motor neurons within the spinal cord on the ventral side. So on the ventral side, where we have nerves that come out the ventral end, this is where motor control is, where motor control resides. So this is illustrated here, a lower motor neuron. Its cell body lives here in the ventral horn, and it would come, its axon would leave the spinal cord, go out the ventral root, and then synapse onto muscles, also glands, which are essentially a specialized muscle, and in order to control the movement of an animal. This is a nice illustration to describe how the difference between afferent and efferent. So we have afferent sensory information. Information is coming away and then being brought into the spinal cord. And then information that's considered afferent information or signals coming into the spinal cord. So this is afferent. Efferent means it's going away from this area. So these are efferent signals that would synapse onto the muscles. Within the ventral horn, there are additional layers of organization. So the neurons that exist in there, those motor units, they're not just in there randomly. They have a map, and that map is specific to whatever layer of the spinal cord that we're talking about. So if we're talking about the more cervical portions of the spinal cord that synapse onto the muscles of the arms and the hand, these units, this is just an example at this level, there's a map there that sort of correlates with the where these muscles are located relative to the body. The muscles that are in the more distal portions of our arms, for instance, say in the hands and in our wrists, those motor units are in the more lateral portions of the ventral horn. Muscles that are more proximal, such as our upper arm, like our deltoid, for instance, those are located more medial within the dorsal horn. And there's essentially a map. It doesn't quite look like an arm. It's not laid out quite just like that. But you can definitely map out that more laterally you're going to find muscle control of distal muscles, and then you're going to find control of proximal muscles more medially. So this is true for the arms. This is also true for the legs. That's going to be where you're going to primarily find this kind of organization within the spinal cord, since those are the two areas of the spinal cord where you're going to find the most complexity of muscle control. So in fact, when you look at the spinal cord and you go through the different layers, you will see that the pattern of the gray matter that exists in there actually varies depending upon the level of complexity of the muscles that are controlled. That just makes sense. So in the cervical portion of our spinal cord, it's relatively larger. And so this is a cross section of what that looks like. And you can see the ventral horn is quite complex here. And that's because these areas of the spinal cord have to control the muscles within the arms. And there's a lot of muscles there, and the muscles of our arms do a lot of pretty extraordinary things. There's a lot of voluntary control, a lot of reflexes, really, really complex. But if you look along the thoracic portions of the spinal cord, the ventral horn is actually quite small. And that's because there are muscles there, some of which are voluntarily controlled. You do sit-ups, you're using your thoracic ventral horn. There are reflexes, of course, that occur there. But there's not a whole lot of complexity of movement that happens for the muscles in the thoracic portion of the body. So the ventral horn there is actually relatively small. Then when you get down into the lumbar portions of the spinal cord, again, we see another enlargement. And you can see that in the lumbar portion and some parts of the sacral portion, that the ventral horn is, again, very, very big. And that's because those motor neurons have to innervate the muscles of the lower limbs and the legs. And again, you have a lot of complexity of behavior that happens for the lower limbs, so you need more neurons here to coordinate all those movements. So whatever the spinal cord looks like when you look at a cross-section, when you're doing a transverse section of the spinal cord, the level of complexity that you see in the ventral horn will be indicative of the kinds of muscles that are innervated. The motor neurons that are within the spinal cord, they can innervate two basic types of muscle. Smooth muscle, which are the kinds of muscle fibers that make up the digestive tract, arteries, glands, for instance. These are all have muscle fibers within them, and these are involuntary control of smooth muscle. And in fact, we're not really going to talk about this much. We will touch on it a little bit when we talk about stress and control of glands, but mostly we're not going to talk about motor control of the digestive tract. Because a lot of that is just kind of involuntary. Instead, most of what we're going to focus on is on the striated muscle groups. The heart is included in there, but skeletal muscle mass, that's what makes up most of the muscles within your body. Those are considered striated muscles, and that's what we're going to mostly focus on. So if you look at a skeletal muscle, so this is in the biceps, and you zoom in, you will see these fibers. And the fibers have this striation pattern, hence striated muscle. And if you look at the portions of the nervous system, since this is a neuroscience class, you will see axons from the CNS, from the spinal cord, synapsing onto the muscle fibers. And they'll have these kind of large globular synapses. And we'll talk about those in the next several slides. So we need to define what a motor unit is, because this is the fundamental unit of how muscle control works. Most skeletal muscles in a mature mammal are innervated by a single alpha motor neuron. These alpha motor neurons, those are the neurons that make up the dorsal horn of the spinal cord, and they will synapse onto one or several muscle fibers. Since muscle fibers outnumber motor neurons, individual motor neurons branch their fibers to synapse on several muscle fibers. There are places where we see motor units that synapse onto a single muscle fiber, but the vast majority of motor units will actually synapse onto multiple muscle fibers. A motor unit, this is referring to a particular alpha motor neuron, so one single motor neuron, and all of the muscle fibers that it innervates. So not just a single muscle fiber, it'll be one motor neuron and all of the fibers that it innervates. These motor units aren't usually sufficient to coordinate muscle activity. So when you're curling and you're getting all buff and you're making your biceps super huge and you're getting all swole, right? That's what the kids say nowadays, I think, right? There are individual alpha motor neurons, individual motor units that are doing that, but they are working together in concert. Your biceps have multiple motor units within it, and they will often work in coordination. This is referred to as a motor neuron pool. So this is a collection of individual motor units that will cause the entire bicep, for instance, if you're doing those curls, that will cause that muscle to contract. And that's because the biceps is made up of a bunch of different muscle fibers, and you have more than just one single neuron that controls that entire muscle. You have a bunch of them, many, and that's referred to as a motor neuron pool. There are three types of motor units, and they have different contractile properties because they're used in different ways. The three types are fast fatigable, that's shown in red, fast fatigue resistant, that's shown in blue, and then slow, which is the green. So let's walk through them. First, to define a few aspects of what we're looking at here. When a muscle contracts, it is exerting force. It is pulling on tendons in order to move the bones in our skeleton. And it can exert force. That's why you can curl weights and get swole. You are using force in order to do that. And we define force as a change in muscle tension in response to a single action potential. And we can measure this out. So you can measure how much force there is, and we can just measure that. One real simple basic physiological way is how many grams can this muscle fiber carry at one time when it starts to initiate. So fast fatigable units, they can exhibit very fast initiation of force when they get initiated. So the initiation time is 0 here. When a fast fatigable unit is activated, it can exhibit a lot of force. So this particular fast fatigable unit is getting up to nearly 50 grams. That's a lot for a single unit. But it starts to fatigue quite quickly. That's why it's fast fatigable. So it peaks and then goes way, way down. Fast fatigue resistant neurons, they have an intermediate force that they will exhibit. So you can see this neuron takes a little bit longer, this whole motor unit, I should say, takes a little bit longer to get up to its peak than the fast fatigable. So it's not as rapid an activation, and it's certainly not as strong. It's about 1/5 of what the other one is. But it's able to maintain its force over a longer period of time. So it can maintain this force up to around 200 milliseconds or so is when it's completely faded, whereas the fast fatigable is fading within about half that time. Then slow units, they do not have a lot of force that they can exert, but they can maintain this for a very, very, very long time. So this is an example of just like a quick impulse that can be initiated with like a single action potential, but we can also look to see how long that it can be maintained. And that's shown over here. And we're changing from milliseconds from a single action potential to what maintenance of muscles can do over time. And that now we can see the scale here is now up to 60 minutes. And you have to pay attention to the scale. Some of you may not be familiar with this. We have a broken x-axis. So we start from 0 to 6 minutes, and all the figures are shown like that. And then after that, we break off because we're skipping over from 6 minutes, going out to 15 minutes, 50 minutes, and 60 minutes. Okay, so you kind of have to pay attention to these break-in axes. So with the fatigability, that's where we can look to see an additional change that occurs over time. So we're pushing these muscle units, these motor units, as much as we can and seeing how long they can maintain their force. So a fast fatigable unit, as you may know, like if you are, say, holding on to a weight in your hand and you put your arm out completely straight, perpendicular to your body, you might be able to hold that out for a minute or two. And it's because you are maintaining and using your fast fatigable units. But eventually they start to tire and you really start to feel sore. And you can see after about a minute, they really start to drop off. And then you can no longer maintain that force way out there. And after about 2 minutes, they are completely fatigued. So they cannot hold up your weight, that weight any longer. So you can hold a lot of weight, but not for very long. So the fast fatigable units, they have a fast forceful response, but it quickly dwindles within about a minute. The fast fatigue resistant neurons, however, they can hold some force for some time. And you can see it's not quite as much as the fast fatigable. But the fast fatigue resistant, they are resistant to fatiguing very quickly, and it can be maintained out over several minutes. In this case, you can see it starting to fade out at 6 minutes. But if you're pushing this neuron, this motor unit, out to 50 minutes, now it's completely fatigued and it can hold barely any force at all. Slow units, They don't hold a lot of force. So you can see the scale here now is only up to 4 grams versus 30 or around 80. But it can maintain this over the time without showing really any fatiguing whatsoever. And you can see past an hour, no difference whatsoever. It's still able to hold 4 grams over the whole time. So these different motor units, we have these different categories because they're used in different kinds of functions of different kinds of motor behaviors. So in this figure, what we're showing is percent maximum force here on some scale that's been normalized up to 100. Could be grams, could be newtons. Those are different units that are used to illustrate force. And then we have percent of the motor neuron pool recruited. So these motor neuron pools, they are mixed in within the spinal cord. And we're talking about the muscles in the lower portions of the spinal cord in this particular example, because we're talking about things that we do with our legs. So we're looking at motor units within the legs. So the slow motor units, we use those most often. Whether you're sitting or standing, you will be using slow motor units because they are maintaining your position. So when you stand, you are using your slow motor units. And you're not applying a whole lot of force when you just stand. The muscles are not doing a whole lot. They're just in a maintained position, but you got to be able to maintain it. So there's some force that's needed, and it's a relatively small percent of the motor neuron pool. It's around 25%. Then when you start to do relatively simple behaviors like running or walking, moving up to running, this is when you start using the fast fatigue resistant motor units. So we can walk for a good long time. You can walk for hours and hours. Running, some of us can run for hours and hours. I can't, but some of us can. And that's because you're generally using your fast fatigue resistant motor units to do that. But then you start to do really complex behaviors, galloping, skipping, jumping, doing box jumps. You know, the CrossFit dudes, they're jumping on these big old boxes, or jumping onto the big old tractor tires and, other crazy things that they do. And that's when you're starting to use your fast fatigable units, and you're really, really pushing your overall motor neuron pool up to the point when you get up to 100% of your motor neuron pool. And that's where the force applied to those is going to be the most. But they will fatigue relatively quickly. You can only do so much jumping onto boxes and jumping onto big old tractor tires. Those are the very strenuous activities, and you can only do it for so long. Individual motor neurons communicate with muscles at a synaptic-like structure that we refer to as the neuromuscular junction. So this is the synapse that is at what is referred to as a motor end plate. What is the motor end plate? Well, a neuromuscular junction, it's a type of synapse, but it's not like a neural synapse. It's a neuromuscular synapse, NMJ. The motor end plate is the postsynaptic side. This is the part of the muscle fiber where the motor neuron axon terminates onto the muscle fibers. Transmission at the neuromuscular junction, this uses the neurotransmitter acetylcholine. We refer to acetylcholine, we abbreviate it as ACH, like this. Acetylcholine acts on cholinergic receptors that are embedded in the muscle fibers at the motor end plate. So this is a typical pattern of what a neuromuscular junction looks like. You'll have the axon come down, and it'll branch, and then you'll have these large presynaptic terminals, and then you'll see a motor end plate underneath. So this is a typical synapse of a neuromuscular junction. An axon, if it has an action potential, the change in membrane potential will move down towards the axon terminal. It'll induce the opening of voltage-gated calcium channels, causing an influx of calcium into the axon terminal, which will act upon the vesicles that are within the axon terminal. These vesicles are filled with acetylcholine. So then the vesicles then they bind to the presynaptic side, releasing acetylcholine into the synapse, and then acetylcholine will then bind to cholinergic receptors. The type of cholinergic receptor we find at the motor end plate are nicotinic cholinergic receptors. The other thing that happens then is that acetylcholine is broken down by another enzyme called acetylcholine esterase, and it breaks it down to acetyl and choline. When acetylcholine binds to the nicotinic receptors, you have a huge release of acetylcholine happening here. That will induce release of calcium within the muscle from the endoplasmic reticulum of the muscle. So you get this huge increase in calcium, and that induces the muscle fibers to contract. We are not going to talk about the contraction of muscles in detail. We're going to focus on the neural side of things. So if you want to learn more about that, consult a biology textbook. There's a little bit of detail in some of the neuro books, so go there. There are all kinds of problems that can happen at the neuromuscular junction that are related to various conditions and diseases. So there's an autoimmune disease referred to as myasthenia gravis. This is a disease that where the immune system attacks the nicotinic acetylcholine receptors that are in the motor end plate. So these receptors are being damaged and they don't work very well. So when a motor neuron fires and releases acetylcholine into the synapse, they can't act on these receptors and you get weakened muscle contraction. Right? If you have something blocking the receptors, then you can't get the release of calcium from the endoplasmic reticulum, and so then you have weakened muscle contraction. But because this is like a constant condition, you will have muscle atrophy occurring within over time. So these muscles, they don't get exercised and they just sort of break down and become very, very weak. It's a pretty terrible condition for people that have it. Curare is an alkaline substance that's taken from plants in South America. There are tribes that have lived in South America for thousands of years. They have used curare as a way of hunting. So they will take it from this plant and put it into poison darts. So this is what poison darts are made of. They'll take some of the poison, put it into the tip of an arrow, shoot it at an animal. The curare will circulate around the animal's body. The curare binds to the acetylcholine esterase receptors. So it blocks the acetylcholine, sorry, not acetylcholine esterase. It blocks the acetylcholine receptors, inhibiting their function. So then when the neuromuscular junction has an increase of acetylcholine, the animal's trying to run away, it's releasing acetylcholine. The acetylcholine cannot bind to the receptors because they're blocked with curare. So then the muscle cannot contract and the animal freezes and then can be killed. That's curare. So you become paralyzed with curare. There are all kinds of neurotoxins that affect the acetylcholine synapse, the neuromuscular junction. We will talk about them in the next couple of slides, but I want to mention two here. One is called tetanus. So that's illustrated here. So with tetanus, what happens with this infection, so it's caused by a bacterium, you get massive release of acetylcholine at the neuromuscular junction. And it cannot be controlled. So you have constant release of acetylcholine at the neuromuscular junction, way too much acetylcholine occurring here, which leads to overstimulation of the muscles where they are contracting all the time. And this guy, this is not like some new hip way of doing piercing, although I'm sure this exists someplace in the world. What this guy is illustrating is lockjaw. So one of the most common symptoms with tetanus is that the muscles controlling the jaw are completely contracted all the time, so you can't open your mouth. So you can't talk, you can just mumble, and eating is really, really hard. So for these individuals, they have to grind up all their food and drink their food through a straw. This can last for a year or longer. Pretty terrible condition. Another type of neurotoxin is referred to as botulism. So this is caused by another bacterium that, so this is showing a can of peas here, because if you ever see a can of food, BPs or anything else that's swollen, that's an indication that there's some bacteria growing in there. And often it's the case that it's this bacterium that is growing inside that food. So if you see a swollen can, do not eat it, throw it out, because it's not good anymore. But if you were to eat it, you would be eating a whole bunch of this bacterium. And what happens with this bacterium is that it blocks the release of acetylcholinesterase. Of acetylcholine. So acetylcholine cannot be released at the synapse. It sits inside the end of the motor neuron, and therefore you get no muscle contraction, which is also not good. This can be used as a tool, though. So Botox is simply the toxin that is produced by this bacterium. And so we can take that and inject it into certain muscles to block the activation of those muscles. So people will use this as a way of dealing with wrinkles on the face. Wrinkles on the face are often induced and are held and maintained because of contraction of muscles within the face. So the muscles are contracting and they're squeezing your skin, and in older people, it looks like wrinkles. If you inject with Botox, you can relax those muscles so that the wrinkles aren't as obvious. Now, of course, this can lead to the condition with bad Botox injections, or if people use it too much, that the muscles of the face are way too relaxed, and we use those muscles of our face to communicate to people. And many times you'll see with those individuals that their facial communication is a little weird. They look a little plastic because they can't actually control the muscles of their face. And that's because their neurons are not able to release acetylcholine. So to go over the steps involved with this process, we have normal acetylcholine that occurs at the neuromuscular junction. It works similarly in neurons as well. That you have acetylcholine released into the synapse. It binds to acetylcholine receptors on the postsynaptic side, inducing some sort of change in the postsynaptic cell. In muscles, it would be an increase in intracellular calcium. Acetylcholine esterase is this critical enzyme that is there to help regulate how much acetylcholine is at the synapse. So you have acetylcholine esterase there to help break down the excess acetylcholine so that you get normal regulation of muscle contraction. You want acetylcholine to be released, activate the muscle, and then stop. So that's what the role of acetylcholine esterase. We have designed all kinds of chemicals that can interfere with how acetylcholine esterase works. One major category of this is referred to as the organophosphates. So the organophosphates, they make up a component of most of the widely used pesticides that are used for insects. So they're the insect acetylcholine esterase, believe it or not, they have the same kinds of neurotransmitters that control muscles there. And they also have acetylcholine esterase, but it's a slightly different kind, given the millions of years of evolution between humans and insects. So we can design certain organophosphates to inhibit insect acetylcholine esterase so that they have too much acetylcholine, getting too much contraction, leading to lethality in the insect. So that's how a lot of organophosphates work. Now, this is the specialization for insects. Of course, there are organophosphates that work on acetylcholine esterase in humans. Those are shown here. So this is just a subset of some of the organophosphates that we know will inhibit acetylcholine esterase in the neuromuscular junction in humans. And some of you may recognize some of these names, in particular sarin, sarin gas, and VX. This was widely used in World War I and World War II as chemical warfare, particularly in World War I. Sarin as well. In fact, this is how chemical warfare works. It's these kinds of chemicals that act as acetylcholine esterase inhibitors. Sarin is sort of notorious for its use in northern Iraq. Saddam Hussein, predating many of my students here, but Saddam Hussein in the 80s had used sarin gas to kill a village of Kurdish people in northern Iraq. And thousands of people died in that attack. Sarin has also been sort of recently used in Syria. So it's still used today. These are nerve agents. They're supposed to be outlawed, but there are plenty of countries that make bad choices and still use them. So what happens, though, when an acetylcholinesterase inhibitor is used in humans? What it leads to is contraction of the pupils. That's one of the most obvious symptoms. Profuse salivation. You have muscles that control saliva release in your mouth. If you have an acetylcholinesterase inhibitor in there, too much acetylcholine getting to those muscles, leading to excess salivation. Obviously, it then acts on the muscles throughout the body, leading to convulsions and seizures. There are muscles that can help control urination and defecation, and you'll get involuntary urination and defecation. And eventually, death occurs via asphyxiation because you lose control over your respiratory muscles, which also use acetylcholine as their neurotransmitter. Really terrible stuff. Not a cheery note, but this is an important component of understanding the connections between how the neuromuscular junction works and how it's been used and abused over the years. Now we're going to walk through the different types of movement and some of the neural circuits that are involved with those with that movement. First, we're going to talk about reflexes. And again, reflexes are rapid, stereotyped, involuntary movements. It's an automated response, and it occurs essentially the same way over and over again. Reflexes. So these are the things that keep us safe, are automated responses because of stimuli that happen in the environment. So they're innate, automatic, difficult to suppress responses. We're going to talk about two examples. One is the knee-jerk reflex. When you were a kid and you went to the doctor for a checkup, the doctor would break out this weird little hammer with a rubber end and would bang on your knee, and your foot would automatically kick up, which was super weird. So it involves tapping a tendon and involves a monosynaptic connection, where that activation releases activates a muscle spindle and also relaxes another muscle. We're going to talk about that in detail in the next slide, but this is the summary of what's going on with the knee-jerk reflex. We will also talk about the flexion withdrawal reflex. So this involves what happens when we have a nociceptive stimulus, pain. This is where pain can be used to have a withdrawal stimulus. So such as stepping on a tack. I don't know about you, but this image gives me a lot of discomfort to see, but many of us have been there and stepped on something sharp. It leads to an automatic withdrawal reflex. And so we're going to talk about the circuit that controls that as well. So there'll be two circuits that we're discussing, the knee-jerk reflex and the flexion withdrawal reflex. So some of what we learned about the proprioception and pain is going to be informative here. The muscle spindles, these are the sensory receptors that are buried deep within the extrafusal muscle fibers, provide proprioceptive information. We learned about those in the somatosensory portion of this course. When a muscle stretches, the spindle stretches, as we learned. This increases the activity of a 1A sensory unit, right? These are these big fat axons that are highly myelinated. And then, so that's when it stretches. And when it contracts, the spindle shortens, and then these afferents decrease their firing. So by pulling on the muscle, it causes this to induce activity. It is driven, obviously, by the activity of motor neurons. So when motor neurons cause these muscles to fire, it causes these muscles to then stretch, which then causes these muscle spindles to stretch, and then that gives proprioceptive information to the brain saying, hey, you know what? The everything seems to be working the way it should. Hopefully you were causing your muscle to move, and sure enough, it's moved, and now you know what position your body is in. That's essentially how that circuitry works. Muscles work in pairs, and this is especially true at joints. At a joint, This is a very simplified schematic of a joint. Imagine this being the elbow. You'll have pairs of muscles, and in a joint, it's referred to as an extensor or a flexor. So the extensor, when it contracts, it will lengthen or open up the two bones that are connected to each other. So imagine your elbow, this would be extending your arm. So when this contracts, it pulls on this end, it's rotating here, and your arm opens up. Flexion is controlled by flexors, which would be causing your arm to be closed more. So this would be bringing your hand up towards your shoulder. There is reciprocal inhibition that allows for this to occur. You can't have both of these contracting at the same time. If you have the flexor contracting, the extensor has to relax. If you have the extensor contracting, the flexor has to relax in order for this to work. There is control within the spinal cord that allows for this to occur. And it is a combination of proprioceptive feedback that allows this to happen. So this is where these muscle spindles come into play. And part of the reason why they have to be so fast is to allow for this coordinated movement to occur quickly. So muscle spindles, if you could imagine in this case, we're trying to cause flexion in this case, in this particular example. So in this case, the muscle spindle, this starts to contract. The muscle fibers start to contract. That pulls on the muscle spindle. So the muscle spindle starts to fire. Okay, so this neuron is firing, it's contracting. This also starts to fire because it's being stretched, and it comes down through here, and this proprioceptive feedback, it goes up to the brain, but it also has collaterals that synapse onto various neurons within the spinal cord. It will connect to an inhibitory neuron that will fire, and then this inhibitory neuron will cause this neuron, it is synapsed onto the opposite muscle, in this case the extensor muscle, the tricep. So this inhibitory neuron is firing, it lowers the rate of firing of this motor neuron so that it relaxes. So when this is firing, causes this to contract, pulling onto the muscle spindle fiber, leading to enhanced input here. So there's sort of a reciprocal kind of connection causing this to continue to fire. But there's an inhibitory neuron that gets activated, which reduces the opposite muscle, in this case, the extensor muscle, allowing the hand to come up towards the shoulder. And then there are muscle spindles on the other side that do the opposite, that would allow for this muscle to relax and for this muscle to contract. The first reflex circuit that we're going to discuss is the knee-jerk reflex. So again, this is this reflex that you would have when you're at the doctor's office. You sit down, you dangle your leg off at the edge of the examination table, and the doctor pulls out this weird rubber mallet and bangs you on the knee. And all of a sudden your leg kicks up. That's, of course, if everything is working. This is to test whether the sensory perception, proprioception in particular, is working properly, if it's connected in the spinal cord correctly, and that if your motor neurons are firing correctly and that your musculature is working correctly. This is a quick, easy diagnostic test, takes maybe 15 seconds, and you can observe the output, and it can tell you an awful lot about whether things are working correctly or not. And if they are not, then you can go on and do additional diagnostic tests to find out specifically what's wrong. But how does this circuit work? Well, let's walk through the different steps. First, the physician will bang on
the ligament that's just below the patella. The patella is the kneecap. Bangs on this ligament. The kneecap is pulled down. This tendon is pulled as well, because it's all connected. And then it stretches this muscle. This is the extensor muscle and the top of your leg, your quadricep. So this gets pulled, and there are muscle spindles that are within here. These are sensory afferents that make up the proprioception network. These muscle spindles, when they are stretched, they induce action potentials in the attached sensory afferent neuron. So you can imagine this muscle spindle is stretched. It causes this neuron that goes through the dorsal root, and this would be in the dorsal root ganglion here, to fire and increase its firing rate. So it starts firing, or at the very least, increasing its firing rate. And then, as a reflex, it synapses onto some neurons that are within the spinal cord itself. It doesn't require any input up to the brain and back from the brain. Instead, this entire circuit exists just within the spinal cord itself. So this neuron, it's increasing its firing rate, and it splits, and it connects to two different neurons. So part of it is synapsed onto a motor neuron that innervates the quadricep itself. So there's a reciprocal connection here. This starts firing, causes this neuron to increase its firing rate. This neuromuscular junction, there's more acetylcholine released there. It acts on the acetylcholine receptors in the muscle, increasing calcium concentrations within the muscle, causing the muscle to contract. The sensory afferent also synapses onto an interneuron that is inhibitory. So this neuron, it increases its firing rate. It is inhibitory, and it synapses onto a different motor neuron that innervates the flexor muscle, the opposite muscle. But because this neuron is increasing its firing rate, it's releasing more GABA here, which is decreasing the firing rate of this neuron. Now this neuron decreases its firing rate. There's less acetylcholine, less calcium in the muscle, and it relaxes. So this contracts, this relaxes, and the foot goes up. So this is an extensor reflex. Causes your leg to extend.
The second reflex we're going to discuss is the flexor withdrawal reflex. This is what's used to withdraw the limb from a painful stimulus, such as a pinprick or, say, touching something like a hot stove. So the flexor withdrawal reflex, what that means is that the limb is being pulled towards the body. That's the flexor stimulus. So the biceps would pull your arm towards your body, and you're not extending out towards the flame. It's being brought towards your chest. And it's all a reflex. You don't even think about it. So in this case, we're looking at the legs. And you have the first part of it is stepping on an attack. And this initiates a nociceptive or pain stimulus. An attack is going to basically activate a single receptive field, maybe a few receptive fields that are really kind of tightly wound right near there. But it's going to be a relatively small portion of the skin that's going to feel it, but it's going to be really, really painful. All of a sudden you feel that, it could be attack. In my case, I've got little kids, so it would be like a Lego piece usually is my life, stepping on a Lego piece. And it'll activate this afferent. This afferent starts to fire. Here's the cell body in the dorsal root ganglion. And this network involves having branching that occurs here that it also breaks off, as we learned in the previous in the somatosensory portion of this course, that these afferents will synapse onto various neurons within the spinal cord. And for the flexion withdrawal reflex, what it does is that it synapses onto excitatory neurons that are within the spinal cord. If you look at this, is like this cross section of the spinal cord, and it's synapsing onto these excitatory neurons that live kind of in the more medial portions of the dorsal horn. And these are local excitatory neurons. They synapse directly onto motor units that make up the flexor portion of our limbs. So these become excited. They both start activating, and all the flexor muscles start to contract, leading to the withdrawal of the affected foot or, say, hand. And so it's a pretty simple circuit with this afferent causing excitation of these excitatory neurons, which then synapse onto the muscles that just make up the flexor. And then within the flexor, of course, if these muscles are contracting, you would then also be activating the muscle spindles, which would cause relaxation of the extensors, right? So that similar circuit that happens for the knee-jerk reflex. But it's initiated here with this pain flexor withdrawal reflex. And it involves no cognition. There's no, you do not need to perceive this consciously. It all happens instinctively, and it's because of this very simple spinal network that does this. Okay, so those are the two reflexes that we have learned about. Those are the two sort of simple and elegant systems. We're going to briefly talk about rhythmic motor patterns and how this occurs in the nervous system. There are all kinds of rhythmic motor patterns that can be induced to allow for changes in behavior. Breathing is one, obviously, but another one that many of us are familiar with is walking. This is a repeated pattern that occurs, and any kind of rhythmic motor pattern that is generated usually involves what's called a central pattern generator, or a CPG. Okay, so that's going to be the key word for the rhythmic motor pattern portion of what we're talking about. The activation of the central pattern generator is often done consciously. You decide that you want to walk, but once it is activated, you don't need to think about it. It produces a rhythmic motor pattern, such as walking and breathing, and you don't need to have sensory input for the most part. And you certainly don't need descending cortical input. You don't need to be thinking about and swinging your left leg and then your right leg. So when biped animals such as humans or quadruped organisms such as cats do locomotion, their limbs are moving through two cycles. There's a stance phase where the limb extends in contact with the ground. So in our case, if you're walking, you have a time when your left leg is in the stance phase and it's extended and it's in contact with the ground, keeping your body up, not falling to the ground. And then you have the swing phase. So then it then switches to a flexed position where you bend your knee and bring it forward to do the next stance phase. And it's a rhythmic pattern because your legs are working in opposite sides. So you do your left leg, then your right leg, and your left leg, and your right leg. You can see in this case, we're looking at what's happening within this cat with the stance and the swing phase. That we have the extensors activated during the stance phase because it's maintaining its position on the ground. And then during the swing phase, flexion happens where the limb has to bend, and and if we were looking at a human, it would swing forward, and so that would be the swing phase. And you can see that they work in opposite, and they just have this rhythmic pattern going back and forth, back and forth. We're not going to talk about the circuits that do this in great detail. This is also something that you don't need to have memorized. But I wanted to illustrate for you a basic circuit that generates alternating activity, just to kind of walk through this. And the way this happens is that it happens at the level of the spinal cord. There'll be input coming from the brain. You're getting a signal from the brain saying, hey, we need to walk. So we're walking. But you're not thinking about the swinging portion. You're not swinging your leg first and then swinging your other leg and then swinging left, swinging right. Instead, you're activating this central pattern generator that creates this pattern for you. And it has to do with reciprocal connections between the excitatory interneurons within the spinal cord that act then on inhibitory neurons, which then act on the opposite excitatory neuron for the extensor portion. So we've got the flexor portion here, and we've got the extensor portion here. This kind of network with excitation, this getting activating this inhibitory neuron, which then quiets this neuron. And then eventually, though, there's activity here that causes this to be activated, causing this to be activated to quiet this. You can see that if this is activated, this is quiet. And then if this is activated, this is quiet. It goes back and forth. And it leads to this pattern of action potentials happening here, then action potentials happening here, while this is quiet and it goes back and forth and back and forth, it then this network will synapse onto the flexors and extensors, leading to this coordinated activity of both of them going back and forth so that you can get rhythmic pattern like walking. And essentially that's how that works. Again, this isn't something that you have to have memorized. This is not a circuit that I expect you to have memorized, but you should know some of the general principles behind central pattern generation and how that works. All right, we're going to finish the rest of this presentation talking about voluntary movements, which are the purposeful, goal-directed, and learned behaviors, things that improve with practice. And there are lots and lots of examples of this. Reading is one. Writing, dance, playing a musical instrument, swinging a baseball bat, those kinds of things. Voluntary motor control now involves the upper motor neurons. So basically everything that we talked about up to this point has been almost entirely just lower motor neuron driven. Everything that's happening within the spinal cord, everything that's happening within the brainstem. We've talked about how initiation events might happen at the motor cortex, but coordinated movement control involves the motor cortex. So the motor cortex is involved with the planning, initiating, and directing of voluntary movements. It is regulated heavily by the basal ganglia. We have a whole lecture on the basal ganglia and the cerebellum. So we're going to hold off on these things for the moment. Right now, instead, we're just going to talk about the organization of the motor cortex and how motor planning occurs within the motor cortex. So first, we've got to talk about how this information gets down into the spinal cord, right? So this information has to be transmitted from the brain down into the spinal cord. There are two broad descending motor pathways that we're going to focus on. There are the lateral pathways. This carries the voluntary movement information that originates in the cortex. So we have the cortical spinal tract and the rubrospinal tract. Just know that they are the lateral pathways. There's also ventral medial pathways within the spinal cord. Ventro because it's in the ventral portion of the spinal cord, and medial, kind of towards the middle. There are 4 of these. These are really important for balance, posture, and locomotion. They help with the controlling of the postural muscles. Many of these fibers originate in the brainstem, and we'll talk about that over a few slides in just a bit. Okay, so first we're going to talk about the cortical spinal tract. This involves one single neuron that goes a long, long distance. There are neurons that exist up in your motor cortex, your primary motor cortex, all the way up here. You get a motor command in the motor cortex. We'll talk about where it's located, but it's in the precentral sulcus in the brain. The fiber passes through what's called the internal capsule. So there's a track in here called the internal capsule. It makes its way into the midbrain, passing through the cerebropeduncle, and then it hits the medulla, which we've talked about before. This is the site of decussation for mechanosensory information. It decussates. Decussation also occurs in the medulla for the motor cortex. So the fibers descend into the medulla, and then they decussate in what's called the medullary pyramid. So this is also referred to as the pyramidal decussation, or the medullary pyramid. So there's decussation that occurs there. So that's when the motor cortex here, we're looking at the right side, ends up on the left side of the body. So like most things in the brain, it is crossed, and it crosses at the medulla. It then makes its way down the cortical spinal tract, which is one of these lateral pathways, and it will then synapse onto the ventral portion of the gray matter within the spinal cord in the ventral horn to synapse onto motor neurons. It can also synapse onto interneurons, and then leading to precise control of the muscles, voluntary control. So again, fibers from the motor cortex pass through the internal capsule, and then you have this pyramidal decussation event where that's where the information crosses at the medulla. If we're talking about decussation in the nervous system, there's a good chance it's happening in the medulla. There are several cortical regions that control and help govern movement. So now we're taking a lateral view of the motor cortex here. Here's the lateral view. Some of these areas are right next to some of the somatosensory areas that we talked about. So there's the central sulcus that we've mentioned as a major landmark, distinguishes the frontal lobe from the parietal lobe. So the parietal lobe is here, and that's where we're going to find all the somatosensory areas. We have the central sulcus, and now we have the frontal lobe. And the somatosensory cortex, shown in green here, is right next to the primary motor cortex, M1. So the primary motor cortex, it's lined up right next to the primary somatosensory cortex. So then we also have premotor areas that are important for initiating the events that are in the motor areas that then cause motor neurons to fire. Those are the neurons that we just talked about in the previous slide. Somatosensory cortex is part of this, though. That proprioceptive information from the somatosensory cortex eventually makes its way to the motor cortex to allow for the coordination of movements. So especially when we're talking about maintaining posture, standing, moving to a sitting position, that perceptive information has to make its way to the motor cortex as well. So the somatosensory cortex figures into this as providing that information to the motor cortex. So one important point is that when we're discussing cortical spinal pathways projecting to the spinal cord, we're referring to the areas in area 4. So that's the primary motor cortex. Premotor area also has a little bit of projection to that as well. The higher cortical areas that help coordinate and plan decision-making around motor inputs, they do not project to the spinal cord. So some of these additional areas, parts of premotor cortex and prefrontal cortex, where there's a lot of decision-making that is involved in making movements, leading to making actions or anticipating actions. A lot of that feeds into the motor input, eventually initiating that movement, but these do not connect to the spinal cord. It's really just these areas that connect to the spinal cord. It shouldn't be too surprising to hear that we also have a homunculus for the motor cortex. We talked about the somatosensory homunculus. We also have a motor cortex that has somatotopic organization. So this is another map within the nervous system. And its map actually matches the somatosensory cortex. It's very, very similar. The lower extremities, our legs, feet, are in the medial portion of the brain. Then we have the trunk. And then we have our arm, which gets a very big representation in the motor cortex since we do so much with our arms. And then we have the face, which also gets a large portion of our motor cortex because we do a lot with the face. So this is what that map looks like. You can see that the face and the hand and the arm has a very large representation. The homunculus for the muscles is very similar to the somatosensory cortex as well, both of which have somatotopy. The body is mapped out in the brain. How do we know that there's a somatotopic map in primary motor cortex? Well, these are old experiments, been done, and this kind of experiment is still used today. We have, we can record activity of muscle units, these motor neurons that are in the somatosensory cortex, and then we can look at the activity of muscles that may be downstream from that using EMG or an electromyograph. So you can use electrodes that will see those muscles contracting, because that is also an electrochemical event. So you can stimulate and record at the same time activity of neurons within the motor cortex, so causing a spike up here in the motor cortex. There's going to be some delay for that signal to get down into the brain, not a terribly long delay, a few milliseconds. And then you can see the activity of whatever motor units are being controlled by that particular neuron. And you can go through the cortex, the motor cortex, and stimulate in here, and you'll see muscles in the upper, like the upper arm, contract. Stimulate right here, you'll see muscles in the hand contract, and you can see the EMG response. You can even see the muscles twitch. Stimulate right here, you will see the face contract. By doing this, you can map out what this somatotaphic map looks like. You can do this. This is showing the hand of a monkey over, I guess, a human, maybe with a really hairy arm. No, it's a monkey, but because a lot of this work has been done in monkeys and macaques. But you can do this in mice, rats, any animal, in order to map out this cortex. Now we're going to talk about how neural activity leads to certain kinds of movements in the primary motor cortex. A lot of these experiments get done with monkeys. They're very intelligent. Their musculature is very similar to humans, so it's pretty translatable. In this particular case, this monkey was trained to move a joystick in the direction when a green light was lit up. So it was given 8 green lights. One light would light up, and then the monkey has to move his hand in that direction. Pretty simple task, and these monkeys can learn it pretty quickly. They were also monitoring the activity of neurons in the motor cortex. So what we're going to look at now is the activity of a single neuron and what happened at different trials within this single neuron. So they're recording from one neuron in the primary motor cortex that seems to be involved with this movement. So they're probably recording somewhere in the upper arm portion of the primary motor cortex. And what you can see, the red line is when the movement occurs. So the monkey is given a signal to say move to the 180 degrees. And then this was the activity that was happening during 5 trials when the light blinked. So this would be trial 1, 2, 3, 4, 5. The monkey was given trials at the 0 degree time point, obviously, and then that would be shown over here, 1, 2, 3, 4, 5. And the red line is when the monkey moved his arm to the right. Okay, the monkey is always moving his arm, but we're looking at the activity of one neuron. This particular neuron seems to be involved and seems preferential to movement to the left. And so you can see when the light was at 180 degrees or 135 degrees, this neuron starts to fire around 100 milliseconds just before the movement actually occurs. So it's firing, then the movement occurs, it starts anyway, and then when the movement finishes, the neuron stops, it decreases its firing rate. It doesn't stop completely. It has a relatively low firing rate, and then when the movement is initiated, the firing rate increases to induce this voluntary movement. And then it decreases when the voluntary movement is done. Interestingly, when you talk about movements to the opposite side of this neuron actually decreases its firing rate to the point where it completely shuts off. So in movements in the opposite direction, this allows the arm to go to the right. So this appears to be probably involved with some kind of reciprocal, similar to the flexor and extensor type network, but instead this is about movement to the left. So this neuron is heavily involved in the arm moving to the left, but it also has to quiet up if the arm is going to be moved to the right. So you'll have a different motor neuron someplace, not being recorded here, where this neuron will then want to fire. That neuron would fire during movements to the right, and then this neuron would then quiet during those movements, this hypothetical right turning neuron. But this is just a single neuron across multiple trials that seems to have preference to the left. So this is a directionally tuned neuron, and we find that many of the neurons within the motor cortex have this kind of directional tuning or specificity of activity that where you can have, it's a voluntary movement, but movements can always go in one direction or the other, and they seem to have this opposite pairing that occurs. So repeating the experiment and looking at different cells show that different populations of primary motor cortical neurons have different preferences. So if you go through and you record the activity of a whole bunch of different neurons, and then you look at the overall vector for what particular neurons prefer, you'll find neurons that will prefer all directions. This monkey he's very talented. He can move his hand in any direction that he wants, and he's going to do it when he's prompted with this green light. If you look at a whole bunch of different neurons, you'll find direction selectivity for all of them. So, so each neuron will have its own preference, and it'll be quiet at the opposite side of it. So you can see for like this particular neuron liking this direction, its vector is primarily in this direction, and you can see it's most quiet in the opposite direction. So you have this direction selectivity for each and every neuron. So that's kind of the initiation of voluntary movements. There's also premotor planning areas that are involved with this as well. So the neurons in the higher cortical regions, in the parietal and frontal lobes, in the premotor area are critical for this planning movement. There are four basic there are three steps to this process. We have the ready, we're paying attention, set, okay, I've thought about what I'm going to do, and then go is now doing it. Excuse me. We can set up the activity of this through various test conditions. Okay, so the ready. So this is a monkey who is given an instruction. There's some sort of instruction, a light or something, or signal that tells it to await instruction of where it's going to move. So it says, okay, something is going to happen. I'm going to have to move here. Set. In this particular case, this monkey knows that there's a series of lights in front of it. So it's been trained, hey, okay, I'm going to have to pay attention to this. The set stimulus is that a light turns on, and he says, okay, so this is going to tell me I have to hit this button, but I will be given an instruction, and in this case, the blue light will turn on to say, okay, now it's time to touch this light. And so the monkey has to attend to each of these three states. So there's the ready phase, the set phase, when it's like, okay, actually, all right, I know what I need to do. I'm going to have to touch this button. And then it's given the go signal, which is the blue light. The activity that we see in the premotor areas of the brain is really interesting with this process. So with the ready phase, there really isn't a whole lot of activity happening in the premotor area. It's being told that it needs to attend, but there's just a certain rate of activity happening. It's not very different from baseline. During the set phase, though, when the monkey is told, hey, you are going to have to press this light, all of a sudden the premotor area starts to fire. So it's firing, and you can vary how long the time is between set and go, but it'll continue to fire during that time. And this is basically telling the brain, hey, this is the motor pattern that you're going to have to do when you do it. Then, when the monkey is given the go signal, and that's the trigger stimulus here, so that would be the blue light turning on, all of a sudden activity in the premotor area stops, and then you would have activity happening in the primary motor cortex, or M1. And so then, so you can see that the premotor areas are involved with this planning process. The monkey being given a signal, he knows he's going to have to touch this, but he's got to wait for the button to hit it. So clearly we have activity related to movement, but without actual movement happening in the brain. We've known about some of these kinds of activity happening for a very long time. And one really interesting specialized kind of neuron that we find in the brain is called mirror neurons. So these are neurons that are within area 6 in the premotor areas. They fire not only when an animal makes a specific movement, but also when the organism observes another organism making the same movement. The way that these were discovered is particularly interesting. So these were first discovered in the 1990s. There was a researcher who was studying movement related activity in the premotor areas. Monkey was trained to say, like, pick up a banana, and it was looking at activity. And then this researcher was changing the thing that the monkey needed to see, that what he was going to pick up. He was going to give it a different thing. And the researcher, you can see this researcher is wearing a lab coat. When he picked up the banana, he saw very similar activity in the same neuron that he was recording from. So whether the monkey did it or if the monkey saw a human or another monkey do it, there would be activity happening within that part of the brain. These are mirror neurons. So they're related to sort of a grasping action in this particular case. And then they also fire when the monkey sees this sort of grasping occur. And it seems to be related to grasping, holding, or moving objects. Let's walk through the different kinds of conditions related to the activity of mirror neurons. So in condition 1, we're recording from this premotor area, and the monkey picks up a nut, and the premotor area starts to fire. Condition 2, the same neuron, we're recording from the same neuron, and this monkey watches another monkey pick up the peanut. It also fires. The same thing occurs when it watches a human pick up the nut. But this is interesting because it's really specific to the hand grasping the peanut. Because when an experimenter uses a very different kind of muscle control, where there isn't a hand grasping a peanut, instead you're using tweezers that are grasping the peanut, the premotor neuron does not fire under these conditions. So it seems to be very specific to these kinds of conditions. We know that there are mirror neurons that are in humans, and we don't just think it, we also know that this occurs. They're not very well characterized, though, because it does involve this sort of very complex process of being able to record. The idea behind what mirror neurons are involved with, it's thought to be that this is how our motor areas and premotor areas observe the movements of others and learn from them. But there's some speculation that they may even be responsible for reading the emotions of others or being used to empathize. So this sense of our sense of empathy and reading the emotions of others obviously has to have some sort of activity within the nervous system. And it simply could be that activity within these areas is also how our body perceives what's happening in another person, including internal or emotional states. There's some relationship, or at least it's thought that maybe dysfunction of these kinds of neurons might contribute to the symptoms of autism, where they have a difficult time understanding the thoughts and intentions and feelings of others. All right, so we're going to briefly go through the descending spinal tracts, the ventral medial pathways that are involved with the control of balance, posture, and locomotion. We're not going to talk about in super great detail, but it's important that you know a little bit about these ventral medial pathways. The ventral medial pathways, they control proximal and axial muscles. They maintain body balance and body posture. So they're controlling muscles within your arms and your legs in order to maintain body posture. These pathways, they include 4 tracts. There's the vestibulospinal tract, the tectospinal tract, pontine reticulospinal tract, and the medullary reticulospinal tract. The vestibulospinal tract, this keeps the head balanced and the shoulders in the proper position as the body moves. It involves the vestibular system, which we learned about in the hearing portion. Head movements are sensed in those semicircular canals and eventually ends up in what's called the vestibular nucleus. So it will sense some sort of movement related to head movement. This tract then splits and allows for bilateral connections to the spinal cord to regulate neck and back muscles to help guide movement. It's a very interesting, complex circuit. We're not going to talk about it in great detail, but that's the vestibulospinal tract. There's also the tectospinal tract, which involves projections from the superior colliculus. So remember, the superior colliculus, as we learned, it receives input from the eyes. That's one big part of what the superior colliculus is important for. But it also has motor output to help regulate control of muscles in the head and shoulders and the eyes. And we're going to learn about that next time. We're going to talk about 2 additional tracts, the pontine and medullary reticulospinal tracts. The reticulospinal tracts, you can break them down into many different subdivisions. They all arise from what's called the reticular formation. So these are a series of complex a meshwork of neurons and fibers that are within the spinal cord. So this is what we're looking at here is this is the spinal cord down here. This is in the brainstem, and we're looking at the underside of the brainstem here. Okay, so like this is the pons, and we have a bunch of different nuclei that run along the length of the brainstem, and that's what we're showing here. There are precerebral reticulatory nuclei, raphe, catecholamine, and other nuclei. We're going to talk about two of these in a little bit of detail, but we're not going to talk about this in any really great detail because it's beyond the scope of this course. In the context of motor control, there are two parts of the reticular formation that provide input. There's the pontine reticulospinal tract. This originates in the pontine reticular formation in the pons, which is an area that's just below the spinal cord. This is involved in anti-gravity reflexes. So what we mean by this is that we have parts of our brain that pay attention to and sense the effects of gravity. Falling is an important thing to resist, and our body has ways of instinctually inducing reflexes to help prevent us from falling. So just maintaining balance, that's going to be regulated by the pontine reticulospinal tract. The medullary reticulospinal tract, originating in the medulla, this helps liberate the anti-gravity muscles from reflex control. So the anti-gravity muscles that help maintain balance to resist gravity, the medullary reticulospinal tract works to sort of counterbalance that. That's the most of what we're going to talk about for the reticulospinal tracts. This is a summary of the descending spinal tracts, talking about some of the voluntary ones that we discussed, as well as a lot of the involuntary ones. I don't expect you to have all of this memorized. There's a lot in here that we haven't discussed in much detail. There's more that we will be discussing in the next time, such as the superior colliculus and its control of eye muscles and control of saccades. But I wanted you to have this information in case you wanted to know a little bit more about how this is organized and where we're talking about the origin of certain things and its overall function. This is just a nice summary of a lot of what we've discussed. Here are the key questions for this lecture. What are the three principal types of movement? What are the differences between upper and lower motor neurons? Where are lower motor neurons located in the spinal cord, and how are they organized with the proximal and distal organization? What is a motor unit? What is a motor pool? What are the three kinds of motor units and what distinguishes them? Fast fatigable, fatigue resistant, and slow. How would you recognize what you're looking at? What kind of motor unit? What is the neuromuscular junction? How does it work? And what are the ways that it can be impaired? Do you know how the reviewed reflexes work? We talked about two of them. What are the circuits that are involved in controlling those? They're relatively simple and kind of elegant. What is a central pattern generator? What is it involved with? How does the cortex control voluntary movement? And what are the mirror neurons? What makes up the cortex? How is it organized? Those are some key, very important questions about all of this. Okay, this lecture's been long enough. We'll see you next time with more about